This module is called Image Data Formats. Up to this point, we've focused on how images are captured, represented, and visualized. In this module, we're going to look at how those images are actually stored on a computer. Image data formats are extremely important in bioimage analysis because they determine how much information is preserved, how large files become, and what metadata can be stored, and which software can then open them. As microscopy datasets continue to grow in size, understanding image formats is becoming increasingly important for both analysis and data management. In this module, we'll address three major questions. First, how do computers store digital images? Second, what are some of the most common image file formats and when should they be used? And third, how do modern file formats and software make it possible to efficiently view and analyze extremely large images? By the end of this session, you should understand how image data are represented in computer memory, recognize the strengths and weaknesses of common image formats, and appreciate some of the technologies that enable large-scale microscopy image viewing. To understand image files, we first need to understand how computers store information. The smallest unit of digital information is a bit, which can have a value of either 0 or 1. A group of 8 bits forms a byte. Computer memory and storage are typically measured in bytes, kilobytes, megabytes, gigabytes, and terabytes. It's important to distinguish between bytes, indicated with a capital B, and bits, indicated with a lowercase b. Image files can become very large because every pixel requires storage space, and modern microscopy datasets may contain millions or even billions of pixels. As image dimensions, bit depth, and number of channels increase, file sizes grow rapidly. When we look at an image, we see cells, tissues, and biological structures. A computer sees something very different. Internally, an image is simply a collection of numerical values arranged in a structured grid. Each pixel location stores one or more values depending on the image type. Software interprets these numbers and converts them into images we view on the screen. Remember that image analysis software ultimately operates on these numerical values rather than on the visual representation itself. One of the biggest challenges in image storage is file size. Compression techniques reduce file size by storing image information more efficiently. There are two major types of compression. Lossless compression reduces file sizes without losing any information. Lossy compression achieves much smaller file sizes, but some information is permanently discarded during the compression process. The slide provides an interesting example. A completely white image measuring 5,000 by 5,000 pixels occupies over 70 megabytes as an uncompressed BMP file. However, because the image contains repetitive information, lossless compression can reduce it dramatically to only a fraction of that size. For scientific image analysis, preserving data integrity is usually critical, which is why lossless formats are often preferred. Here we see a visual example of lossy compression. The original image is shown along with enlarged views before and after compression. At first glance, the compressed image may appear very similar to the original. However, when magnified, compression artifacts become visible. Fine details may be blurred, edges may become distorted, and subtle intensity variations can be altered. These differences may seem minor for casual viewing, but then they introduce serious problems during quantitative image analysis. This is why lossy formats should generally be avoided for storing primary microscopy data. Lossy compression not only changes the appearance of an image, it also changes the underlying pixel values. One way to observe this is by examining the image histogram. Because pixel intensities are modified during compression, the distribution of pixel values changes as well. This means statistics thresholding results in quantitative measurements may differ from the original image. The key message is that lossy compression can alter both what we see and what we measure. In this exercise, you'll compare images saved in multiple file formats. First step is to open images from previous exercises and save them using different formats in ImageJ. Then compare the resulting files. Consider questions such as, do the images look different? Have file sizes changed? Has any metadata been preserved or lost? Are these images still suitable for quantitative analysis? This exercise highlights the trade-offs between compatibility, storage efficiency, and data preservation. I'll pause here so you can have a chance to do this exercise by yourself. I'll now demonstrate how we can perform these steps in QuPath. So the first step is to open the image samples in ImageJ and then save them in different file formats. So from QuPath, we can go to Show ImageJ, open our samples. We'll start with fluorescent cells. Here we can then save as, and save it as a JPEG file. We'll select it, a location where we can find it afterwards. And then we'll go to our File Explorer and we'll change the viewing settings to Details. If you're not able to see the suffix of image files in Windows, you can change this to Show File Name Extensions. Sometimes you might also want to consider viewing hidden items. Here we can see that we have a JPEG format of our fluorescent cells and a TIFF, and that the size of these is quite dramatic between the two of them. When we now open our JPEG file in QuPath, we can see if there are any differences in the image itself. One way we can change how we view images is by having one image alongside another for easy comparison. So I have on my left the JPEG, and by right-clicking, multi-view, and set grid size to 1:2, we now have the viewer split in two. You can see that when I click on the image, a red boundary forms around it for the one that I've selected. We can then select the right viewer, and now that has a red boundary around it. We'll then open our TIFF into that. We'll first make sure that synchronize viewers is off. Make sure we have reset the viewer to capture the whole image, and then we can now synchronize views, where we can then zoom into specific parts of the image and see if there are any differences between them. Here we can see that maybe the value of some pixels are different using the overlay in the bottom right. If you don't see the overlay in the bottom right, then we can go to the icon of a monitor screen and make sure that it's toggled on. In this case, Show Cursor Location has been toggled on. We also have the option for the scale bar and also the Show the Slide Overview, which is in the top right of the viewer. Let's compare several common image formats. BMP files are simple and widely readable, but generally uncompressed, resulting in large file sizes. JPEG files use lossy compression and are excellent for sharing images or presentations, but they may lose detail and alter pixel values. PNG files use lossless compression and are widely supported, making them a good choice for figures and graphics. However, they are generally limited to RGB representations and are less flexible for microscopy metadata. TIFF is one of the most important formats in bioimaging. TIFF, or Tagged Image File Format, supports a variety of compression methods, preserves metadata, and can store high-quality image data suitable for analysis. As a result, TIFF and related microscopy formats are often preferred for storing scientific image data. TIFF is one of the most widely used image formats in microscopy. However, TIFF files can vary substantially in how information is stored internally. To improve interoperability between instruments and software, the bioimaging community developed OME-TIFF. OME stands for Open Microscopy Environment. OME-TIFF combines image data with standardized metadata that describe experimental details, image dimensions, channels, spatial calibration, and other important information. This standardization makes it easier to share data across software platforms and research groups. As microscopy datasets continue to grow, even OME-TIFF can present challenges. Researchers are now routinely generating datasets that are hundreds of gigabytes or even multiple terabytes in size. OME-Zarr is an emerging community standard designed specifically for cloud-compatible image storage. Rather than storing all image information as a single large file, OME-Zarr organizes data into smaller chunks that can be accessed independently. This makes it important for multiple users to remotely access only the portions of data they need without downloading an entire dataset. Large microscopy images create unique technical challenges. A whole slide image may contain billions of pixels, far exceeding the amount of memory available on a typical computer. Specialized file formats address this problem by organizing image data into smaller regions and multiple resolutions. Rather than loading entire image into memory, software can retrieve only the information required for the current view. This allows users to navigate very large images smoothly and efficiently. A useful analogy is Google Maps. When you open Google Maps, the application does not download the entire world at full resolution. Instead, it loads only the portions of data you are currently viewing. Large microscopy image platforms use similar strategies. These include lazy loading, where data are only loaded when needed, chunking, where images are divided into smaller pieces for efficient access, and resolution pyramids, where different versions of the image exist at different resolutions. We saw how this was a setting in importing images within QuPath, and together these approaches allow software such as QuPath to interact with extremely large images while maintaining good performance. Let's review what we've learned. We discussed how computers represent digital image information using bits and bytes. We explored common file formats and their relative strengths and weaknesses. Finally, we introduced modern microscopy file formats and the computational techniques that make large image datasets practical to store, share, and analyze. These concepts are essential for selecting appropriate file formats and building reproducible image analysis workflows. Digital images are ultimately stored as data composed of bits and bytes. To conclude, remember that every digital image and microscopy datasets can be extremely large, and so compression plays an important role in reducing storage requirements. However, it is critical to understand the difference between lossless compression, which preserves data, and lossy compression, which permanently alters the image. Different image formats serve different purposes. Formats such as JPEG are useful for sharing and presentation, whereas TIFF-based formats are generally preferred for scientific image analysis. OME-TIFF has become a widely adopted standard for storing microscopy images, along with their associated metadata, while OME-Zarr is helping addressing the challenges of cloud-scale bioimaging. Finally, technologies such as chunking, lazy loading, and resolution pyramids allow softwares like QuPath to efficiently navigate images that would otherwise be too large to load into memory. Understanding these concepts will help you choose appropriate file formats, preserve image quality, and build scalable image analysis workflows. That brings us to the end of this module, and I hope you'll join us for the next module on annotating images.